All right, so welcome back to part 2 of chapter 15. We are going to talk real quick about two different types of DNA repair. We're just going to cover two just to give you an idea of some of the different types of repair that occur out there. There are some other forms, but these are the two most common, so we're gonna talk about these. So DNA repair is a necessary part of DNA replication. DNA repair also takes place outside of replication after transcription, simply because the DNA is being kind of messed with, and we have extra enzymes that are going to come in and they're going to kind of read the DNA and make sure that the nucleotides are formed properly, there's no alterations to the nucleotides, that the base pairs are correct, it's A to T, C to G, there's no changes in the structure, and that the sugar phosphate backbone also remains intact, so there's no weak spots. Now, in order for DNA repair to take place, first off, we have to be able to detect that damage is even present. And then enzymes that are responsible for repair will move in and actually carry out that process. So the type of repair, the types of repair that we're going to talk about are alterations in direct repair. In direct repair, an enzyme is going to remove a modification. So either a methyl group, an alkyl group, or some kind of chemical modification has occurred to the actual nucleotide itself. So it's going to go in, it's going to remove the modification and repair it. When we have alteration to a DNA strand, we actually remove a section. Let's say there's even just a single base that is incorrectly base paired, or it's twisted, or something is wrong with it. In that repair, the enzyme is not going to just remove that base and repair it. It's going to remove several bases on either side of it. So it's really going to remove a small section and then rebuild that section properly. Now, the two most common that occur that we're going to talk about are nucleotide excision repair and methyl-directed mismatch repair. I believe the chapter in your textbook may have a few others mentioned, and you can certainly take a look at those, but for brevity, we're going to simply talk about nucleotide excision and methyl-directed. So in NER, nucleotide excision repair, this is the most common type of repair that's going to occur. And there are several different types of mutations or damage that nucleotide excision repair can cover. Here on the right is an example of a thymine dimer. Thymine dimers occur when two thymines that are next to each other, instead of base pairing with their complementary adenines, they get structurally altered. Usually UV light is a common form of thymine dimer formation, and they'll end up binding to each other and next to each other instead of to their complement base pair, and you can see that we get this kind of bump here in the actual sugar phosphate backbone. This is sometimes referred to as a nick. And this nick or bump that we see in the DNA here is what is detected by our enzymes that are responsible for proofreading the DNA. It should be nice and smooth, and so it moves along and it says, hey, wait a minute, there's a bump here, what's going on? And it then stops and carries out nucleotide excision repair. So the region that's encompassing the nucleotides, a whole region, you can see here in this diagram, here is our a complex right here. This is a light excision repair, and it's detecting the bump here. It's detecting that there's damage. So after the damage is detected, these enzymes, the A's are going to be released. This B part of the enzyme will just simply stick to where the damage is for location, and then we're going to have the C portion of this enzyme now attach. And notice it's going to cut upstream and downstream several bases from the actual damage itself. When this is removed, because the thymine dimers are detected in that strand, there's another enzyme here that's going to move along that actually breaks the hydrogen bonds between all of these bases on both sides of where that repair occurred. Now, in excision repair, we're going to have the undamaged strand is going to remain behind. That's going to serve as the template for the new strand that's coming in repair. Nucleotide excision repair takes place in both eukaryotes and prokaryotic organisms.
Nucleotide excision repair is an important general process that cells use to correct many different types of DNA damage, including UV-induced thymine dimers. Although present in all eukaryotes and prokaryotes, the molecular mechanism of this system is best understood in prokaryotes. This animation will illustrate the process of thymine dimer repair in E. coli. The nucleotide excision repair process uses a set of proteins called UVRA, UVRB, UVRC, and UVRD. UVR stands for ultraviolet light repair of pyrimidine dimers. But remember that this system is also used to repair other types of DNA damage. In addition, DNA polymerase and DNA ligase are used to complete the repair. The process begins with the loading of a protein trimer onto the DNA double helix. This trimer consists of 2 UVRA molecules and 1 UVRB molecule. This trimer moves along the DNA, scanning for damage that distorts the double helix. When the trimer encounters DNA damage such as a thymine dimer, it stops. The UVRB protein remains at the damaged site, while the 2 UVRA proteins are released. UVRC then binds to the site and makes 2 cuts on the damaged strand. Both cuts are several bases away from the damaged site. UVRD, a helicase, then binds to the site and separates the segment of damaged strand from the rest of the DNA molecule. UVRB, C, and D are then all released from the damaged region. This leaves a gap that is filled with the proper nucleotides by DNA polymerase and then sealed by DNA ligase. The end result is that the damaged DNA has been replaced with the correct bases.
So you can see there a key component of the nucleotide excision repair is that even though the damaged area is very small, the enzymes remove and replace several bases both upstream and downstream, and this is really to ensure enough room for all of these enzymes. We got to make sure that DNA polymerase can get in there and carry out that replication process, as well as ensure there's no possible damage in that immediate area, that the damage hasn't spread out. Now, the other form of repair that we're going to talk about is methyl-directed mismatch repair. Here, this is where a base mismatch is where we don't have our A to T or G to C rule, right? So we have a set of bases that are not matching, they're not complementary to each other, so they're not base pairing properly. This occurs when DNA polymerase makes a mistake. Usually, more often than not, DNA polymerase will correct its own mistake. The number of hydrogen bonds that occur between improper base pairs it causes the DNA to not form properly, and DNA polymerase 3 will catch that during replication. It'll literally stop, back up, remove a bunch of bases, and then start over again. So DNA polymerase, in addition to its synthesis ability in building new strands during replication, DNA polymerase 3 also has its own proofreading and repairability. So it makes a mistake, but it'll correct itself because it is also a proofreading enzyme. In methyl-directed mismatch repair, this is found in all species, both prokaryotic and eukaryotic. It detects the mismatch and will remove a segment. You can see here in step 2 here, we had this repair going on, and we have an incorrect base, I think right here. Here we have the incorrect base pairing, and so all of this section here gets removed. And then it will get, DNA polymerase will come back and fill in that region, and now we have the correct base pairing occur. In this case, it's going to detect the mismatch, and it's going to remove the segment just like we saw in nucleotide excision repair, but it's going to specifically repair the new strand and not the parent template strand. Because remember, the parent strand was correct. It knows the parent strand was correct. So it's not going to remove the parent strand. I mean, this is going on during replication. So it's not going to remove the parent strand. It's only going to remove the strand that was being synthesized, because that is the strand where the mutation has occurred. Notice the G was originally present. This G was originally present on the parent strand, so it's only going to remove the daughter strand, the section of daughter strand, and fix it.
Although rare, incorporation of base pair mismatches during DNA replication does occur. There are DNA repair mechanisms that can fix such mistakes. One such mechanism is the methyl-directed mismatch repair system. In Escherichia coli, this repair system can detect a mismatch shortly after DNA replication and repair the newly synthesized strand, thus preserving the base pairing that was present in the original DNA molecule. The system relies on the fact that methylation of a newly synthesized DNA strand does not occur until several minutes after the strand is made. This provides a window of time during which the newly synthesized daughter strand in a DNA double helix can be distinguished from the parental strand, which is already methylated. A sequence of DNA with only one methylated strand is said to be hemimethylated. Actual recognition of a base pair mismatch is the role of the mut-S protein. Mut-S binds to a mismatch site and forms a complex with another protein, mut-L. The formation of this complex recruits mut-H to the region. Mut-H binds to a hemimethylated site. The mut-S, mut-L complex interacts with mut-H by a DNA looping mechanism. Mut-H then makes a cut specifically in the non-methylated strand. An exonuclease digests the non-methylated strand from the cleavage site to just beyond the mismatch site. This leaves a gap in the daughter strand, which is filled by DNA polymerase III and then sealed by DNA ligase. The end result is that the mismatch has been replaced with the correct base pair as indicated by the original DNA strand.
So that is methyl-directed mismatch repair. And again, it's important to remember this is occurring during replication. And this is going to cause that loop to occur so they can move backwards and digest. That nuclease can go back and digest the daughter strand, not the parent strand. So that brings us to the concept of cancer, and cancer is a cell mutation gone bad. So cells go through mutations, and the more mutations a cell develops over time, the closer to cancer it can become. Now, some mutations will result immediately in cell death, and most of our cells, or not most of our cells, but most of us have had some type of cancer cell in our system at some time or another, but our immune system overcomes it. And our immune system says, hey, you're not behaving properly, you're not doing what you're supposed to be doing, and the immune system will get rid of it. So the actual development and diagnosis of cancer, even early on, is considered a failure of the immune system, meaning the immune system did not catch it, did not get rid of it ahead of time, and so this cancer got out of hand before the immune system could do anything about it. About 1 million Americans are diagnosed with some form of cancer every year, and about just under 50% of them will actually die from the disease they're diagnosed with. And about 10% of cancers, people have a higher predisposition to develop the disease because they have inherited some trait that has made them more susceptible. And most cancers do not involve genetic changes that are passed from parent to offspring. So 90% and above of the cancers that are developed are due to induced mutations, not because of inheritance or germline mutations. Carcinogens are environmental factors that are known or linked to the development of cancer after exposure. So about 80% of all cancers are related to some type of carcinogen exposure. This would be things like cigarette smoke, sunlight, UV light, chemicals, anything that causes some type of mutation in the genetic material when exposed to it, and ultimately can lead to genetic changes in somatic cells. DNA alterations lead to changes in gene expression. So again, some of those mutations that we talked about in part 1 can occur, and this can alter, usually like missense mutations for the most part, different point mutations. This can either stop the production of a protein, meaning, or I shouldn't say stop production, but this could either cause loss of transcription, loss of translation, or loss of function due to misfolding. And this can occur in both structural and non-structural genes. So cancers will originate from a single cell that has gone through some type of mutation. And the mutation, depending on where it is and what type of gene it's in, that mutation allows the cell to grow either at an abnormal rate or to have abnormal phenotypic expression. A tumor is when other cells surrounding the original mutated cell also begin to have changes in their genetic expression, just as the original cell did. So the original cell kind of tells the other cells, hey, we need to start replicating. So a tumor is an overgrowth of cells, and the tumor has absolutely no purpose. It can be benign or it can be precancerous. A benign tumor is one in which cells are just not doing what they're supposed to do, but it's not cancerous. It's not causing any trouble. It just doesn't really do anything. So benign tumors can either be left alone or they can be removed. If a tumor is considered precancerous, this means that the pathologist has seen changes in the cellular structure or morphology, or even in the cell expression, that makes the doctor kind of wonder, something's not right here. This level is off or that level is off, depending on where the cancer is, and they may want to keep an eye on it. If a cell enters or a tumor enters into a cancerous stage, stage 1 is malignancy. So in malignancy, the tumor starts to grow. And one of the primary characteristics of cancers is unregulated cell growth, meaning the cells just start dividing and increasing in population. Once this tumor starts to kind of reach out, it'll become abnormally shaped. Some cancers will have tentacle-like structures to them, the tumors will, and they'll start invading and attaching into tissue, and so they'll start invading into healthy tissue. So this is an invasive stage, or stage 2, or stage 3. And then in the last stage of cancer, we enter what's called the metastatic stage. And in metastasis, this is when cancer, the tumors become very large, unsustainably big, or fragile. They can become very fragile. And some of the mutated or cancerous cells will break off or slough off of the tumor, enter into lymphatic tissue, get filtered into the lymph, and then move to other parts of the body. The cancer starts to move. And when those cells get lodged in other parts of the body, they will be expressing rapid growth signals and cancerous type signals that will tell surrounding cells to start doing the same thing that it was doing before. And so now we start seeing tumor formation in other parts of the body. If left untreated, cells that are malignant are going to eventually cause death. It's going to invade tissue and cause organ failure. So here we have some lung cancer here. Here is our benign growth, and this is just kind of our initial mutated cell, but then other cells around it start growing, and you can see they're abnormally shaped. They're very tight and very close together. Large tumors are kind of like a thick rubber ball. Then in metastasis, we start to see that cells are breaking off. These tumors become very large, oftentimes very fragile, and so they start sloughing off and moving around. We can see here on the left is a tumor. This right here is a tumor inside of a lung, and this here is a healthy lung. So you can see how it's caused massive inflammation, fluid, and of course all of this tumor in here has no purpose and it's non-functional. So it starts to reach up into other parts of the tissue, and you can see this tumor here is obviously going to disrupt the function of the lung. So there are two types of genes that are most commonly the cancer-causing genes, or the genes that get mutated that are associated with cancer. The first is the proto-oncogene, and the proto-oncogene is a gene that is associated with cell proliferation or cell growth. Now, proto-oncogenes are genes that are usually turned off. They are used very early in development, in utero or in fetal development, when cells need to divide very, very rapidly. So when proto-oncogenes get turned on, they send out really intense cell proliferation signals to surrounding cells, and these cells will begin dividing so quickly that DNA polymerase doesn't necessarily have the ability to proofread like it should. And this can induce further mutations from there. And so these cells start to rapidly mutate. Now, tumor suppressor genes are kind of the opposite. Tumor suppressor genes are always on, and tumor suppressor genes are part of the regulation of cell growth. So in this case, these genes are usually turned on, and they force cells to keep kind of some personal space. For lack of a better term, keep them in line so that they don't grow really super close together. Now, in cancer, in tumors, those cells they grow really tight in these big lumps, right, in a tumor formation. But this tumor suppressor genes normally prevent cancer. So if we get some type of mutation within the actual tumor suppressor gene, it can no longer suppress tumor formation. It can no longer downregulate cell proliferation signals. So we see a cell that can now kind of grow out of control. It's not being regulated. So a proto-oncogene, usually there are the four most common changes genetically that occur to proto-oncogenes are missense mutations. That's number one. We see gene amplifications. So in this case, the gene gets amplified. It gets upregulated. So there would be a mutation in the enhancer or in the repressor. Or silencer in the silencer sequence. Then we have chromosomal translocations. So this would be like a transposon moving into usually the regulatory region of a proto-oncogene. And then we have what are called retroviral insertions. Retroviral insertions are retroviruses. Somebody gets a viral infection and it is a retrovirus. Now retroviruses when they enter into a cell, they introduce RNA and they reverse transcribe their RNA into DNA and then insert that DNA into the genome of the cell. When retroviruses do this, many retroviruses will insert in the regulatory region of a proto-oncogene. And if they do this, then the regulatory region is no longer the same sequence. It's no longer recognized by a silencer, so the gene will then be turned on. So oncogenes are genes that encode for cell growth. They encode for these growth factor hormones that induce mitosis and increase what's referred to as cell proliferation or increase in cell production. They bind to the oncogene products. The products of oncogenes will bind to a cell surface and lead and go through a cascade, a cell signaling cascade that tells the cell to divide. So if these oncogenes, once a proto-oncogene has been mutated, it's now referred to as an oncogene. And so the products of these oncogenes bind to other cells, surrounding cells, and tell them you need to divide and you need to divide now. And they tell them to divide very intensely. It's a very strong division signal. This will cause the cell to go through division to start DNA replication processes and forward itself through the cell cycle into the mitotic phase too quickly. And this allows for other mutations to build up. So mutations in genes that produce cell growth signaling change the oncogenes into producing super high levels of these proteins, and this signal becomes very intense. Oncogenes promote cancer by basically keeping the proliferation pathway on all the time. It just flips on the on switch and holds it up there and just says divide, 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 divide. And so these cells keep dividing. Now remember, when cells divide, they copy their DNA first. So if the oncogene is mutated in the original cell, every daughter cell from that cell division is also going to contain that mutation. So ultimately, they're all giving off this cell proliferation signal, and that cell proliferation signal can become very intense pretty quickly. Sometimes in cancers, the amount of gene product is abnormally high, and this is sometimes used as a biomarker to detect early, hopefully detect cancer very early. Sometimes the gene produces, the mutation results in a gene product which the protein itself is hyperactive. So it's not that the gene is turned on and being transcribed more quickly, but it's that the actual protein works better than it did before the mutation. Missense mutations and chemical mutagens are usually what lead to these types of mutations in cancer. And again, in a proto-oncogene, many times those mutations are either going to be in the gene itself, or a lot of times they're in the promoter region or in the regulatory region of it. Here's just another visual of that change. So this G got changed to a T, and we went from a glycine to a valine. So this is another missense mutation. In gene amplifications, we can get an abnormal increase in the number of copies of mRNA, and this is going to cause too much of the protein to be produced. For example, what's meant by gene amplification is transposons, transpositions, or during replication, RNA polymerase is rushing, is moving through, and it makes 3 copies of a gene instead of 1. If I get 3 copies of the gene, that means if we have transcription factors, if we turn on this particular gene, and it's an oncogene, we're going to transcribe 3 times the amount of gene product, right? It's going to make 3 times the amount of gene product that the cell actually needs, because we now have 3 copies of the gene instead of 1. Chromosomal translocations can do things like break a chromosome. They can take a piece of chromosome from one area and move it to another. So in one case, we have the breaking of 2 different chromosomes, and the ends of those chromosomes will fuse together. So that can happen right here. That's what they're showing you right here. And because in eukaryotic cells, our chromosomes are linear. Bacteria do not have that issue. Their DNA is circular. In some types of chromosomal translocations, these are found in tumors depending on where those translations occur. We have here a fused chimeric gene, and this is where we have a chromosomal translocation either from some other part of the chromosome into this region of this particular gene, or from a completely different chromosome. Chimeric genes is what we're looking at here, and so we have a piece of gene from somewhere else in the chromosome being fused to this existing gene right here, this proto-oncogene, and now this completely changes how this gene is going to be expressed. And then, of course, here are the retroviral insertions, and you can see here are viral sequences. And the virus inserted its DNA sequence right here, in this case, in upstream of the promoter. Viruses cause cancer because they either disrupt the regulation of proto-oncogenes and tumor suppressor genes, or they can actually bring genes of their own. So we didn't really talk about transformation in this particular case, where we have viruses will grab genetic material from a previous host accidentally, and then they bring it with them and insert it into the next recipient host. This can occur. This is a process known as transduction in bacteria. And viruses can sometimes bring actual oncogenes with them in their infection, and when they insert into the host DNA, those oncogenes will get expressed. Most cancers are going to be caused by mutagens. There's a couple of viruses. Hepatitis B is the most highly associated with liver cancer. We see Helicobacter pylori is a bacteria, actually, that can that is associated with gastrointestinal cancers. It's the same bacteria that causes ulcers. HIV is associated with some Burkitt's lymphoma and a few other cancers as well. Sometimes, human papillomavirus, that was the other one, HPV, which is what the Gardasil vaccine is against. So Gardasil was considered a cancer vaccine not because it acted directly on mutations of cells, but because it protects you from the human papillomavirus, or HPV. If you don't get HPV, then you're not going to develop HPV-associated cervical cancer. That's the idea behind that. Some viruses can, again, carry cancer-modifying genes, and then some will carry oncogenes in their own genome and then introduce those into the host when they insert into the genome. Next are the tumor suppressor genes, and tumor suppressor genes are supposed to prevent cancer. They're supposed to prevent the formation of tumors and slow down and highly regulate cell proliferation pathways. Proteins that are encoded by tumor suppressor genes usually have one of two functions. They can maintain by monitoring and repairing alterations. They are often associated, are sometimes called checkpoint proteins. They are during the cell cycle. When we go through the cell cycle, we go from like G1 to G2 to M to S phase. Every time a cell moves from one region of the cell cycle to the next, they're going to encounter what are called CDKs, or cyclin-dependent kinases. And these CDKs are check proteins. These proteins are basically going to proofread the DNA and make sure that all of the base pairs are properly matched. And they're just going to kind of maintain the integrity of the genome. So these genes need to be on all of the time so that the DNA is constantly being checked. Of course, if we have disruption of one of these genes, then some of these checkpoints will get missed. If checkpoints get missed, then we're going to start seeing mutations beginning to accumulate. There are other tumor suppressor genes that encode for proteins that negatively regulate or inhibit the cell proliferation pathways. And so they're going to stop cell division. They're going to say, hey, no, you don't need to divide right now, so that cell division doesn't get out of control. So tumor suppressor genes encode for proteins involved in cell cycle. Our this video is not working, so we'll have to post that one for you. So how do tumor suppressor genes lose their ability to regulate the cell cycle and to ensure that our DNA is properly base paired? The three most common ways is for a mutation to occur within the actual gene itself. That's, like a missense, even a insertion deletion. This is going to cause the actual gene product to no longer function, so we lose a checkpoint. The chromosome in which the gene is located may be missing, or it carries the one that carries the tumor suppressor genes may be missing. So this would be a chromosomal disorder. And then we have abnormal methylation. So remember a few lectures ago I talked to you about in the regulatory regions, like in the operator or in the very beginning of many genes, we had regions called CpG islands. These are GC-rich regions, and the reason they're important is because G binds to C with three hydrogen bonds. This means that when I have a region of DNA that's very GC-rich, that DNA is harder to break apart. And genes that are normally turned off will have really high CpG regions. If they're not methylated properly, then these regions can get turned on or off, depends on what's going on at that promoter. So abnormal methylation of those islands near the promoter regions can sometimes disrupt the transcription of tumor suppressor genes, and so they won't get transcribed. Because they get highly methylated, if there's a CpG island present there, it's like permanently turning off the gene. So checkpoint proteins produced by tumor suppressor genes are called cyclins and cyclin-dependent kinases, or CDKs. And they are, again, they are like the roadblock. A cell cannot advance from one phase of the cell cycle to the next without first being checked by these CDKs. the formation of what are called cyclin CDK complexes are oftentimes stopped by checkpoint proteins. And there's one in particular that's really important, and that is P53. P53 is one of the most important proteins in the cell cycle, and it is kind of the one that decides whether a cell finally, it's that last point, whether or not the cell can finally move into the M phase or mitosis. About 50% of all human cancers are associated with some type of mutation in the P53 gene. So P53 is in our G1, right? It's in our G1 checkpoint. If P53 is expressed, its gene is going to be induced when damage is encountered. So if expression of the gene of P53 is induced when DNA is damaged, it's going to halt the cell cycle. It's going to stop everything. In this case, the cell cannot move on from G1 to S. S is the synthesis phase. This is when DNA replication occurs. Oops, cation. If the DNA gets repaired, and what P53 is doing is it's stopping the cell cycle and it's going, wait a minute, wait a minute, wait a minute. We've got a mutation. There's a mistake here. We need repair. We need the repair groups to come in and check this before you can move on. So if the DNA is repaired, P53 will allow it to move into the S phase for DNA synthesis, for DNA replication. But if it doesn't get repaired, then P53 is going to bring along a nuclease and the cell's going to go into apoptosis, which is programmed cell death. So if the damage is too severe, it can't be repaired, the cell is going to go into what's called apoptosis, or programmed cell death. Apoptosis is the cell telling itself or being told by another cell, hey, you are not functioning properly, you got to go. Caspases are enzymes that function as proteases, and what will happen is P53 will trigger caspases, and those caspases will start to run around the cell and start turning everything off. They do this to induce cell death. That's the beginning of apoptosis. For multicellular organisms such as ourselves, it's really important that we just simply kill an occasional cell with cancer as opposed to allowing that cell to divide. We're going to kill a single cell as opposed to allowing that cell to divide uncontrollably and eventually perhaps disrupting or destroying an organ, which would be much more detrimental to the organism than a single cell that can be replaced. Checkpoint genes can sometimes be rendered inactive because of this mutation. And so the division of healthy cells doesn't necessarily get affected, but the division of unhealthy cells will. So there's an example here of mice that are missing the P53 gene. So they point mutated the gene for P53 in these mice so that it was non-functional. Cell division led to normal growth, was regulated correctly, but checkpoint proteins are not necessarily. So P53, what they were showing is that P53 is not necessary for normal cell development. P53 is about checking. So as long as there were no mutations, then the cells would divide and the organism would function properly. However, the mice required much less exposure to mutagens for the development of cancer than mice that still had intact P53 genes. So this loss of checkpoint proteins is really a big deal in cancers. Cancers will get rid of these checkpoint proteins so that cells can divide uncontrollably, is essentially what they're doing. So cancer requires more than one mutation most of the time. It's going to require multiple changes to the exact same cell. These are usually kind of like dominoes. Once one occurs, oftentimes multiple other mutations will begin to occur as well. Also, since most cancers are due to mutagens and exposure, those mutagens will oftentimes cause more than one mutation. It usually starts with some kind of benign mutation, but over time, additional mutations, especially if something like P53 is missing or not functioning properly, then we can see how those mutations will build up over time. When cells become malignant, it becomes much more difficult to treat those cancers and to target them. So that is our lecture on mutations, repair mechanisms, and just a little bit about cancer and how cancer develops and where it comes from, the different types of cancers, the two most common types of genes, human genes that are involved in cancer development. This concludes all of chapter, I believe, 15, and I will see you in class. Have a good night.